Hello classmate, my name is Lee Ji-hyun and I'm in the plant immunology lab. I'm going to talk about what I'm learning in the lab. At first, I'm going to tell you why plant immunology is needed and why they're competitive in the future market. And then I'm going to tell you what plant immunology and finally, what kind of skills I'm using in the lab. First, I will tell you why I study plant immunology. Look at the picture on the left. A train is trying to transport food. This train looks like it's hard to keep up with the rapid population growth. Statistics predict that the population by 2050 will reach about 10 billion. But as you can see from the picture on the right, food production on Earth is hard to keep up with this population growth. Eventually, there will be food shortage. And so far, many people argue that food is a distribution problem, because on one side, people die of obesity, and on the other side, they die of hunger. Ironically, even under this situation, some people are trying to make biofuels with foods such as corn. Food shortage appears to be a national problem in growing population, poor distribution, and continued attempt to manufacture biofuels. To solve this problem, we need to increase food production. Statistics show that 60% of food production can solve this problem. Among the many ways to increase food production, the concern of our laboratory is to reduce the loss of food it produces. Food losses from pathogen and insect amount of 1/5 of total output, and in countries like developing countries, about 50% of the total loss is caused by pathogen. So, what are the ways to overcome this problem? The triangle on the right is called disease triangle, which describes the relationship between the factors that affect the occurrence of plant disease, including the pathogen, the environment, the host. Even if you can cut down on stock of the pathogen here, you can reduce the food losses caused by pathogen. Second, I will tell you what plant immunity is. The elements that cause disease in plant can be largely divided into abiotic stress and biotic stress. Here's an example of that. So, how do plants protect themselves from all these different elements? Plant defense are divided largely structure and biological defense. Here's an example of that. Among them, I'm studying the innate immune defense of plants that protect themselves with their own immunity as pathogens enter. Let me explain the innate immune response of plant. First, when pathogens break into plant from outside, and plants use their pattern recognition receptor to recognize the preserved pattern of pathogen, we call these PAMs. And then plant transmit plant signal, and finally, plant make the immune response like the HR reaction. This is called the pattern triggered immunity, PTI. But in order to overcome this reaction, pathogens are also able to evolve a weapon called an effector and inject it into the plant. This effector has some effect on the protein of plants so that plants do not recognize pathogen. We call this is effector triggered susceptibility, ETS. To overcome this, plants make resistance protein that recognize effector directly or indirectly and protect themselves. This is called effector-triggered immunity, ETI, and it is stronger and lasts longer than PTI. Next, I'm going to talk about how to study this plant immunology. So, before I tell you, this is one of the projects that I'm working on. Viral A is an effector of a human pathogen called Shigella that invade the human body and cause disease, and is known to combine directly with autophagy to prevent air-to-gauze trafficking and recycling, and interact with a protein called calpain to cause necrosis or apoptosis. There's a study that shows that viral A causes disease in real plant, and I'm studying how viral A interact with the plant protein that cause some reaction. As a way of studying this, the first is to hybrid. This is a method of dividing protein that binds to carforgine by binding site and activation site, and fusion your interesting protein each, and then expressing it inside the yeast. If they are strongly expressed, the reporter gene that exists behind the carfor binding site will be strongly expressed, which will allow them to produce amino acid essential for yeast or excal, so that they can grow on medium with specific composition. On the right side, things are factor I use. The difficulty of this experiment is if self-activation of binding domain take place, it is difficult to accurately measure the strength of the protein that interact, and this is self-activation depends on the gene. In order to find the protein that you are curious about whether you are interacting or not, you need to do screening. Using hybrid from the previous slide, we put a high concentration binding site domain into the library that has a large number of activation site domain, and we find the gene that interact with. This method is to check binding between protein. And this is what I used is strain information. Now we have found that genes interact within the yeast, but whether they interact in real plants is another question. If you want to know if they are actually expressing and interacting with plants, you have Western blot and CoIP that you can check the amount of protein. But there's also a BIFC that you can see with your eyes. BIFC is bimolecular fluorescence complementation. The principle of BIFC is divided YFP into N-term and C-term and connect each target gene to the N-term and C-term and express in plants. You can observe the YFP with fluorescence microscope, and the stronger the interaction, the stronger the light will be. This is what I use factor for BIFC. This is the process of doing BIFC, which first infiltrated the agar into tobacco and then observed the result 3 days later. The advantage of this experiment is that you can visualize the interaction of protein. However, the downside is that the signal can be arbitrarily emphasized to control intensity, so you should be careful about it. This is what I prepare for seminar class. Thank you for listening.